Science of Genomic Integrity. Talking about stem cells and transit amplifying cells and how with not too many divisions of the original stem cell, you can get this large population of intermediate cells that eventually in normal tissue give rise to post-mitotic, fully differentiated cells. But this is also a way in a cancer situation, if these stem cells are mutated, to then get a large population of similarly mutated cells to create the tumor. We saw that under, at least using colon cancer as our model, most stem cells seem to be sort of somehow protected in the colonic epithelium. They're protected at the bottom of these colonic crypts, which are sort of plugged with mucus, which helps keep potentially carcinogenic molecules away from them, things like that. And then these transit amplifying cells eventually sort of keep moving up out of this crypt and up into the villus, to make the functional tissue, and you can sort of watch that happen in the cells in some way. We talked a little bit about how stem cells might be replaced, especially given the sort of normal situation where after a stem cell divides, one of the daughter cells remains a stem cell and the other becomes sort of that first generation transit amplifying cell. But several models exist where you can convert backwards. So the transit amplifying cell, presumably an early one, could convert back to a stem cell, or you get symmetric division instead of asymmetric division, something like that. Just one example. And that kind of shifts over into looking at some aspects of mutational repair, although we're going to look more in detail at that today. But this reminds us about the proofreading activity of DNA polymerases. And that if you mutate that, your mutation rate goes up, and the survival in mice that have this mutation goes down. This is going to be sort of part of our intro for today in terms of being able to repair certain types of mutations by cutting out the offending section and re-synthesizing that section. We're going to spend some time looking at that today. And then we kind of spent most of the last class looking at different types of mutations and kind of what causes them. So we saw that there are endogenous types of mutational events just kind of because. So every once in a while, a guanine nucleotide spontaneously hydrolyzes off the nucleotide, and that sets up a purinic site. We saw spontaneous deaminations can change one nucleotide base into another, or change it into a different chemical that has different base pairing properties, was sort of the theme that we saw there. We get spontaneous oxidation events from reactive oxygen species, just from the fact that we do aerobic metabolism. And again, that can change the base pairing capabilities of certain bases. And then there are the exogenous types of scenarios like ultraviolet light, and they can cause thymine dimers, alkylating mutagens that add alkyl groups like this methyl group, which again can change base pairing characteristics. Sometimes it's larger things that start out sort of innocuous, but through the cytochrome P450 family of enzymes that are supposed to get rid of toxins or detoxify things, sometimes they make some compounds worse. We saw that. And then sometimes those worse things, again, add on to bases, very often guanine, and that messes that up. What else do we see? Eating certain nasty types of fungus or inhaling them can cause problems, because some of them have toxins that, again, can add on to nucleotides, again, often guanine, and cause problems. Cooking your red meat at too high a temperature on the grill creates heterocyclic amines, which can be problematic. And I think that's where we got to. So there are lots of potential issues, both within our own cells and coming at them sort of from the outside. So naturally, well, at least I think sort of naturally, the cell has over time evolved mechanisms to repair many of these types of problems, but realize, of course, that no biological system is 100% effective. Right? right? So while we have a lot of systems in place, none of them can possibly be expected to work at 100% accuracy. Plus, as we'll start seeing, and hopefully you've been thinking about already, these would be key genes, the genes that code for these repair mechanisms, would be key features. If I were a cancer cell, I'd love to get rid of those, right? Because that would increase the mutation rate, which of course is a fundamental property of pushing towards the multi-hit model and progressing towards worse and worse phenotypes in terms of cancer. So those are sort of going to be our two big themes for today. So we'll look at some of these basic mechanisms. Again, we're not going to look at them in any huge mechanistic detail. If you like that kind of stuff, then I recommend BMB 400, Dr. Ingersoll's class, if you want the nitty-gritty details of these. We're going to, again, look at them sort of more generally. So, one general mechanism that we have against UV light, especially, is, of course, melanin production by melanocytes in your skin. So that's certainly a mechanism that should decrease the ability of UV light to penetrate your skin and therefore cause thymine dimers. So it's fairly effective against UV light to an extent, of course. There is no biological mechanism, though, for other types of radiation, like X-rays or cosmic radiation, for the most part, presumably because there's no selective pressure for that, because most of those kinds of radiations are kept out through the atmosphere. So presumably, if those things reached the surface of the planet, we would have evolved something. But it does make you think about possibilities for, once you leave the atmosphere and you leave the planet, okay, that's kind of a formidable thing to think about if we're actually going to send people off to Mars and off to places even further out. Is how do we protect them from those kinds of radiations for which we have no biological protection. Okay, so that needs to be somehow built into those spaceships or whatever, because that could be a problem. We're going to have a series of sort of single enzymes that can fix certain things. Some of those oxidative damages that we've seen from reactive oxygen species can be sort of directly fixed by things for enzymes called superoxide dismutase and catalase, can often take care of a lot of those issues, hopefully before they cause damage, ideally. And so if you can take that, reactive oxygen species and through one or both of these enzymes convert it into water, for example, or back into oxygen gas, okay, you're going to hopefully take care of the problem before it has, caused a problem. Okay? So these are often mutated, as you might expect. And then there are other non-protein-based things. We always hear about antioxidants, vitamin C, vitamin E, those kinds of things. Those aren't proteins, but those are considered sort of molecular scavenger types of molecules that can, again, hopefully, get rid of those reactive oxygen species before they do damage. What they're showing us here in this figure is glutathione S-transferase. Which connects glutathione, which is a small sort of tripeptide, that the enzyme will attach to various electrophilic compounds, which many carcinogens fall into that category. And if you do that, it's supposed to sort of detoxify them into something that, again, can be worked on further and further reduced to detoxify them. This particular gene for this enzyme is particularly sensitive to promoter methylation. So we see that the promoter for this gene is often highly methylated in different kinds of cancers. So we've seen that sort of mechanism before as, again, a higher than background mutation rate, so instead of mutating it, you just kind of turn it off. So that keeps those kinds of molecules sort of toxic in that sense, and therefore more prone to cause mutations. There are a series of dealkylating, oh, sorry, yes?
Why are some genes in particular more prone to methylation of promoters than others? Is there any particular reason for it?
I don't know that they're particularly more prone, but for whatever reason, that is one in particular that the book pointed out is, shown to be highly methylated in a lot of cancers. So it might just, ultimately all of this is random, right? So it's just that we see it more so, but perhaps it's more effective on some, for some promoters than others. Would be like general answer. There are a series of dealkylating enzymes, so if alkylating agents are bad, if we can pull them back off, that should be a good thing. So there are a number of these types of enzymes. So if this compound has that alkyl group, this particular G product, MGMT, can pull that back off, and then you're back to normal binding again. No foul, or no harm, no foul kind of idea. This is showing mice with lymphoma. The percentage goes up if you knock out that particular dealkylating gene. So the rates of mice with lymphoma without that particular dealkylating enzyme goes up. And there are others as well with it. Epoxyaldehyde can do it as well. So there are a number of enzymes sort of in that category also. Kind of the interesting thing though here, sort of interesting little aside, is that while certainly effective, right, this is a good anti-mutational kind of thing, sometimes these same enzymes can muck up the works of certain chemotherapeutic agents. So in some cases, the cancer has sort of a love-hate relationship with some of these kinds of things. So in some cases, under certain circumstances, still having this, or in some cases, even increasing some of these dealkylating enzymes turns out to be pro-cancer because they'll detoxify the chemotherapeutic agents that you're trying to use to kill the cancer. So it's an interesting balance that some cancers have sort of struck with some of these repair enzymes. Okay, so there are some enzymes that are trying to sort of take care of the problem before there's a problem. Melanin, these superoxide dismutases, glutathione oxidases. There are some that are trying to reverse, sort of directly reverse the damage. Right? You put the alkyl group on, if you take it back off, you're good to go. And then there's another category, sort of a broad category of repair after the fact. Okay, so some kind of mutational event has happened, and can we fix it in sort of a different way? And this is sort of the type that we saw earlier in the chapter, where there is some change that causes some sort of distortion in the helix. And then there are enzymes or proteins that are able to recognize sort of various levels to this. So this first example here is showing a small change that doesn't distort the helix hugely, but enough that it can be seen. So think maybe, maybe instead of this sort of larger alkyl group, it's just an extra methyl group or something like that. Will that distort the helix? Ever so slightly. So there is a mechanism for that. So there are proteins that will recognize that distortion, hone in on that messed up base, and the one highlighted there in yellow, okay, and remove that base. Okay, so that's done by a family of enzymes called DNA glycosylases. So they are going to lyse off the base from the sugar. That's what that means. But essentially, what then have we created? We have a missing base. What do we call that? Not yet. Right. it's an AP site, either an apurinic or an apyridinic, whatever. This creates an AP site. Okay? We don't like AP sites, right, because there's a missing base, so if this were trying to be transcribed or copied at this moment, that would be bad, right, because the enzyme wouldn't know what to do with that, so we need to fix that. So that's really no better than that, in a sense, right? In fact, it might be a little worse if that's where it stops. So we can't stop here, so we don't. So there are sort of two alternatives. This is relatively new information. So kind of two alternatives to this. So how do we fix that one missing base that was pulled off by the repair mechanism? Well, apparently there's a short patch and a long patch way to fix this. The short patch is just to cleave out that one nucleotide. Okay, so they are doing it sort of piece by piece. They pull off the base first, then they pull out the rest of the nucleotide. There's just one missing. So the short way is just that one back, okay, and then ligate it together. So there is a DNA polymerase that will do that, and DNA ligase makes the phosphodiester bond, and then presumably, if all goes well, then you're back to normal. The long patch repair is going to be more similar to what we're going to see next, where they don't just repair that one base, they sort of yank out the next few bases and repair a stretch, including the one that they originally removed, but a few extra, and then they ligate it together. So this is the short patch version, I think, is the new information. We always thought it was always a longer fix. So there are specific proteins, sort of what I usually call surveillance proteins, that are kind of looking for these distortions in the helix, and these are looking for sort of small distortions. But we've seen others that would end up distorting the helix much more significantly. Think aflatoxin, okay, or some of those polycyclic amines that are adding much larger groups to these bases, so you're going to distort the helix much more significantly. There are different surveillance proteins that are going to look at that level, or be able to see, quote unquote, that level of distortion. And then essentially the rest of the story is much like this long patch repair. So they find that distortion, instead of just dealing with the one problematic nucleotide, they just kind of jump ahead, cut out a stretch on either side to get this sort of large gap, and then get DNA polymerase along and resynthesize that section, glue it together with ligase, and hopefully problem solved. So this is more of this kind of sort of the standard way of doing this. So only under very small circumstances with, again, just a small distortion, they try to fix just the one base. Most of the time, they just cut out the offending section and resynthesize. So this is called nucleotide excision repair, where they're excising a larger stretch. So those are sort of the ideal ways. There are circumstances, though, where the cell is just, it's kind of so overwhelmed with problems that it resorts to using what have become known as error-prone polymerases. And here, and how the cell kind of chooses which one of these to do is still a little bit unclear, I think. But here, what it tries to do, especially in the situation with thymine dimers, and especially if there are lots of them, okay, if there's just one thymine dimer, then it's probably going to fix it this way. But if there are a large number of these thymine dimers, especially, it reverts to this error-prone repair process where it tries to use the complementary or the homologous chromosome to fix this section. And that, as you might imagine, becomes a much more error-prone process in that they have to try and find the homologous chromosome, line it up perfectly, make sure they're using the correct strand as the new template strands. There are just lots of problems associated with this, but sometimes it works. and sometimes it doesn't. So it's very, very much error prone, and it is really considered kind of a last-ditch effort of the cell to fix the mutation. Presumably, like if this were happening, kind of in a non-cancer cell, if they ended up, most likely, if they ended up not being able to fix it, or this sometimes makes things worse, hopefully then P53 gets involved and says, all right, we've tried, we did due diligence, it's time to kill this cell. But of course, in the cancer situation, probably by this point, P53 is long gone, and we don't have that, sort of that circuit to kill the cell. So this does exist, kind of surprisingly, but it must do, it must do some level of good under certain circumstances, or presumably we would have evolved this away by now. So it must do some level of good, but sometimes it does additional damage. This is the way that it tries to fix things. So those are some sort of fundamental ways that cells have to repair, generally speaking, repair some of these types of problems that we're looking at in this chapter. Mutations then in these types of genes cause problems. And one great example of this shown here is the scenario called xeroderma pigmentosum, XP for short. See if I can spell this correctly. Is a problem in nucleotide excision repair, and it makes these individuals particularly sensitive to UV light, which causes thymine dimers with the specific mutations. They don't get fixed properly, so rates of skin cancer in these individuals is much, much higher than they are in the general population who have these fixed, or have these functional repair enzymes. So you can see here, people with XP have a much, much higher incidence of skin cancer than the normal population. So it has been linked to, like I said, a mutation in mismatch repair, in excision repair. But it turns out that there are a number of steps in that process, as we saw. So a mutation in kind of any of those steps leads to this, or can lead to this problem. So that's what this figure is trying to show. I don't know how well it shows that. But it shows sort of the genetic approach that figured this out. But the upshot of this is that there's not just a single gene that causes this problem. There's a sort of a cluster of genes, all presumably involved in this sort of excision repair pathway. So it might be, in some people, it might be the enzyme that recognizes it, and other people it might be the enzyme that that excises the problem. So there are a number of what are called complementation groups if you're looking at this through the lens of a geneticist. But the major theme here is that there are multiple genes associated with this particular syndrome. And there are others. So there's not just XP, there are a number of different cancer syndromes that have been linked to or mapped to mutations in specific repair genes. And this table lists some of those, or some relatively common examples. So in mismatch repair enzymes, nucleotide excision repair, double-strand breaks, which we haven't really talked much about yet, non-homologous end joining, we'll get to that in a second. And then sometimes it's more about the DNA polymerases, so helicase and replication, because that's part of the repair process, right, to resynthesize that stretch. So if there's a mutation in the particular polymerase that does that step, that could be a problem as well. And I think we looked at Leigh-Fermeli syndrome in some previous chapter. So that's part of DNA damage alarm proteins, those are things that are looking for various damages. And so there are certainly syndromes associated with repair in these, or I'm sorry, mutation in these repair enzymes. And this one is Oh, this is part of the problem in hereditary non-polyposis colon cancer. Is a mutation in the gene for this particular receptor that gives us an early stop codon. And so it's a nonsense mutation, and you can only get a portion of the receptor, and that just gets degraded because this would never get inserted into the membrane because it's before the membrane spanning section. So that's part of the sort of signaling cascade leading to microsatellite instability and other types of problems, which is another example. And then to put all this again back into the cancer context, here are, again, genes that have been inactivated in human cancers that are involved in various mismatch repair steps. So, as we were saying before, from a cancer point of view, getting rid of these genes is a good thing, right? They're going to increase the cancer, or increase the mutation rate because we're not fixing them. And upping the mutation rate means you're going to up the chances of getting rid of getting rid of P53, getting rid of RB, getting, you know, active mutations in RAS and all the many other examples that we've been looking at. So getting rid of repair mechanisms is a very pro-cancer type of phenomenon. And this is probably some specific example. Yeah, this is the MLH1 gene, which is not in this list, but MHL3 is, again, this is a gene that's methylated and inactivated in endometrial cancer. And others that you might be familiar with are the BRCA genes, BRCA1 and 2, that were isolated first in breast cancer. And these turn out to be parts of repair processes as well. They both bring other repair proteins to sites of problems, is what this slide is supposed to be showing us. And so if they get mutated, then there can be less of a sort of signal for all of these repair proteins to sort of congregate and fix specific problems. And the loss of BRCA2 especially has been correlated with all sorts of chromosomal level craziness. So if you look where all the arrows are, these are chromosomes that are starting to fuse together, and only small pieces or small fragments of chromosomes. You know, there's a deletion here of some kind. You know, so you get all sorts of weird karyotypic problems because of some kinds of mutations that are not getting repaired properly. And eventually, this tends to be part of this phenomenon called non-homologous end joining, which is sort of similar to the idea of that breakage fusion rebreakage syndrome that we saw when you start losing telomeres. Well, it's the same kind of idea. If you start breaking chromosomes sort of randomly along their length, there are no telomeres that are protecting them, so they start fusing together, and we can get non-homologous, right? So non-homologous chromosomes. Their ends start joining together, and then that starts causing these weird karyotypic problems. And then think about if you have two pieces of two non-homologous chromosomes now attached to two different centromeres, and they're being pulled to two different sides of the cell, the dividing cell. That just creates new breaks, which creates new fusions, or creates a new round of end joining. So this, coupled with the breakage fusion breakage cycle that we've already seen with telomeres, associated with telomeres, but again, thematically, these are kind of the same idea, can start creating this genomic level, sort of craziness in terms of the karyotype. And that's what these couple of slides are just showing, that the homologous ends, non-homologous ends start fusing together as, again, an attempt by the cell to try and fix things. Right, so it knows that these just random ends are not supposed to be there, and in sort of a last-ditch attempt to start fixing things, you just start, okay, random ends are bad, let's link them together so at least the ends are gone, but Obviously, in most cases, that tends to cause a lot of problems because chromosome 1 and chromosome 17 are really not supposed to be linked together, right? So often that causes that level of problem. Again, under relatively normal situations, that cell would die. But again, if this is part of this cancer phenomenon and cancer progression, these crazy genotypes are going to be pro-cancer, okay, in the sense that mutation rates are higher, and you're doing all kinds of other things, and randomly, maybe fusing chromosome 1 and chromosome 17 uses two genes that together, gives you a phenotype that again is pro-cancer. We've seen that before. It's kind of a weird picture that's trying to show, I think, this phenomenon where different non-homologous chromosomes are being fused together. It's kind of a weird way to show that, but that's what that's kind of showing, and they've seen it in certain types of cancer. So again, don't worry about the specifics, but this phenomenon has been seen to happen. And this phenomenon is new to the book, new to this edition of the book, so if you have a first edition, you might want to find and read this little section. And this is our big new word for today. This word is chromothripsis. Use that at dinner tonight. Your parents will say, "Oh, I'm glad we sent you. You can stay out." From two Greek words, as all really some good scientific words come. Chromo, right? Chromosome, same idea. Chromo, colored, because we can stain the chromosomes to see them. The Greek word thrips means to break or to shatter. So this is literally chromosome shattering. Not completely understood. This is pretty new, but it is literally that. So that chromosome starts shattering into little tiny pieces, and then that starts this sort of cycling. Okay? The cell goes nuts. The cell says, "Oh my God, I have all these random ends. We need to fix this," and it will just start randomly joining them together. At least in this example, it's all from the same chromosome, so it's not exactly non-homologous end joining, but it's homologous, random homologous end joining, I guess, and puts it back together in crazy connotations, in crazy ways. So clearly that chromosome is not really going to be overly functional in that new configuration, and then sometimes not all the pieces are put back together, so that those pieces are going to be lost, and especially if they don't have centromeres. So people are still kind of working out the mechanisms for this in terms of really what's causing this in the first place, and how exactly the cell is kind of coping with trying to put the pieces back together. But as we all know from the nursery rhyme, once Humpty Dumpty gets shattered too much, you really can't put it back together again.
So based on A there, does this happen only on the P arm, on the short arm, or
Oh no, I'm sure this is happening.
according to those lines?
Right, but I'm guessing this is a specific example of a more generalized phenomenon. Okay, so here's your nice word for today. Yes, sir.
The idea that it's it's a very violent mutation of the chromosome just breaking apart. What ideas of what causes such a thing to occur?
If they know, I don't. If anybody knows, I don't. Like I said, this was new to this edition of the book, and they basically say, "We've seen this happen. We're working on why." The repair part's easy. That's just this. But what's causing a nice chromosome to almost literally explode or shatter into little tiny pieces will be an interesting mechanism, whatever it is. Yeah, so as far as I know, people don't know. Okay, that's out there too, so that's really going to mess up the karyotype. And then all this, or at least some of this karyotypic or genotypic level of change can of course include changes to chromosome numbers. So this is looking at sort of normal tissue versus cancerous tissue just at chromosome 8. So if you're looking at just random normal cells, the vast majority of them are going to have 2 copies of chromosome 8 with, maybe those with 4 probably mean that those are actively dividing cells, right? And perhaps those ones are sperm cells or egg cells, something like that. But if you look at cancer situations, you're going to get kind of the whole gamut. So within a tumor, there are going to be cells still with 2 copies, although they're kind of in the minority, and others with 3, 4, 5, 6, 7, up through 11 copies of chromosome 8 in this particular example. So we think back to other classes and chromosome number problems. At this point, this would still be, even though it's a lot of wrong numbers, this would be aneuploidy, literally not the true chromosome number, 3/3 there. So obviously this is going to change copy number of genes and therefore expression levels of genes, and we've seen that issue already, where some genes get amplified, to high copy number, and too much of a good thing is a bad thing. Nick is probably one of our best examples of that. And then this is showing a phenomenon called just instability, so chromosomal level instability versus microsatellite instability. And from what most studies suggest is that these phenomena are sort of mutually exclusive. So chromosome level, so if you have things at the chromosome level causing aneuploidy, then the cell usually isn't at the same time mucking around with microsatellites, one of those little stretches of repetitive DNA. But if they're if the cell is in the midst of microsatellite instability, chromosome number is usually kind of stable. So for whatever reason, they seem mutually exclusive phenomena. That's not too big a deal for us. And then these last couple of slides are showing sort of another side to all of this that I don't think we often think about, which is mutations in genes that are coding for aspects of cell division itself, genes that code for pieces of the mitotic spindle. So think about that. If you're mucking up the mitotic spindle or kinetochore proteins at the centromere and things like that, that's certainly going to change the very nature of how those chromosomes are moving during cell division, which could certainly lead to things like that one, but where is it? There it is. To multiple copies of the same chromosome going to one cell and none going to the other, that kind of idea. So that can certainly lead to things like aneuploidy. And these are just kind of experimental micrographs showing different aspects of it. So things like spindle here, or this chromosome has 2 separate this one chromatin has 2 separate centromeres. So it's trying to be pulled to both of them, and it's not actually going anywhere, so that's staying in the middle of the cell. So when the cell divides, what happens to that one chromosome? Does it get lost to both? Does it end up going to one and not the other? Others have shown mutations in genes that are coding for the centrosome, which is the microtubule organizing center. So that could have much more global changes to the entire mitotic apparatus. And then occasionally problems in the apparatus can lead to, instead of it dividing into 2 cells, it divides into 3 cells, and of course that's going to radically change chromosome number and things like that. So this is an aspect that, like I said, I don't think we often think about, are mutations to the mitotic apparatus. Could certainly change chromosome number and change genomic level karyotypes. So there are a number of genes that have been shown now to be either mutated, methylated, so turned off, or in some cases overexpressed, that somehow change chromosomal stability, presumably during mitosis. So again, don't need to memorize the table or anything, but there are some good examples in this sort of category that we don't consider very often. Couldn't do it. So there's a lot of potential problems out there. There are a goodly number of ways to